Hello everyone, Dr. Alex Vasquez here to let you know that I'm working on the next video in the Human Microbiome and Dysbiosis in Clinical Disease series. This next video will be on the gut-brain axis, focusing more on solutions, and this will be delivered in 2 parts of 2 hours each. So please keep an eye out in your mailbox, your email inbox that is, for the notice of this new video, which again should be available pretty soon, hopefully this weekend, possibly next weekend, as I'm still making some edits to that. And again, this is going to focus on the gut-brain axis and especially focusing in on solutions to that, at times, rather complex interaction and dynamic. So again, keep an eye out for this in your email inbox. It'll be coming from healthythinking.substack.com. Thank you very much. Hello everybody, Dr. Alex Vasquez here with the last video in our series on human microbiome and dysbiosis in clinical disease. Today I will cover part 2 of gut-brain connection and microbiome-mind interactions. Part 1, I discussed the introduction and some structural concepts. I'll review a few of those again today. We'll talk about some specific gut-brain mechanisms again in this conversation, which is mostly going to focus on clinical implications and interventions. Then I'll conclude with a quick summary of perhaps some of the more important concepts. For this section on the gut-brain axis, I have outlined the following teaching and learning objectives. First of all, of course, we always want to start with a survey of the literature so that we have a good understanding of the pathophysiology involved, especially for the conditions that I'm going to focus on, which are listed here. After that, the main focus of any continuing medical education is to change clinical action. So for that reason, of course, I'm going to focus on and discuss and review the evidence base for general interventions and, where appropriate, also very specific interventions. So the task today is actually quite large. I think that teaching and writing is generally pretty difficult. Good and excellent writing and teaching are even more difficult. And that is when we're talking about things that are well understood. So you can imagine the complexity of talking about things that aren't well understood, such as dysbiosis and neuroinflammation, and those, of course, will be the major topics for today. I've certainly done my due diligence in that I've read hundreds of articles on this topic, including essentially all of the major review papers. I'll talk about some of the better papers, perhaps, later in today's presentation. All of these have been published in leading journals, and not a single one of these articles has included all of the information that I'm going to cover today. As I state towards the bottom, the task of scientists is to discover facts and relationships. The task of intellectuals is to amalgamate all of that information into functional, comprehensible, and impactful models. To understand the role of neuroinflammation in dysbiosis-induced disease, one must first understand neuroinflammation. And then to understand, again, the role of neuroinflammation in dysbiosis-induced disease, one must understand dysbiosis. If you want to connect those two together, then I think we have the opportunity to gain some unique insights into common conditions, such as depression, anxiety, persistent pain, including, of course, fibromyalgia, chronic fatigue syndrome, and also complex regional pain syndrome. I'll talk about all of those today. So this is the second half of the gut-brain presentation. Let's talk about some of the main components of neuroinflammation. This includes the microglia or microglia. These are the immune cells within the brain and the spinal cord. Those are sensitive to pathogen-associated molecular patterns called PAMP, and also DAMP, which stands for damage-associated molecular patterns. That includes ATP and mitochondrial DNA from mitochondrial dysfunction. When the microglia are activated, those trigger the astrocytes to engage in a state of activation as well. Those astrocytes subsequently release glutamate, and that, of course, affects neurotransmission because glutamate is a major excitatory neurotransmitter within the brain and spinal cord and also in the periphery. When those neurons become activated by microglia activating the astrocytes, then that, of course, has clinical consequences because when those neurons are more activated, especially at the glutaminergic NMDA receptor, that appears to correlate with not simply brain inflammation specifically, but clinically anxiety, depression, and persistent pain, and also expedited neurodegeneration. Anytime the microglia get activated, they're going to release more cytokines and more free radicals, and those are going to impair mitochondria. When the mitochondria get impaired, that's going to reduce the ability of those neurons to deal with the increased metabolic demand placed on them by increased glutaminergic neurotransmission.